Hello, sophomores. Welcome to your first screencastic video notes. I'm going to go very quickly through this. I'm not going to read the slides to you, but if it's something that I think you should pause and take notes on, I will say that. Otherwise, I'm going to try to only add the extra information that you would be getting if I were talking to you in class. So read this slide. You need to know everything that's highlighted. This first slide shows you a traditional scientific method. This should be old news. I know that my students started learning about this in about second grade, or my kids, so hopefully this is all review. However, I am going to add some fancier words that I think as a 10th grade student you need to know the definitions of. So here we go. Just kidding. Here we go. Why won't this work? All right, here's a list of vocabulary. As you go through the slides, you should be able to define these. Pause right now, write these things down. You need to know and understand and be able to identify everything on this page. So this is an outline of your goals for this PowerPoint. Hardest thing for kids to understand is the difference between a law and a theory. Basically, theories are what I teach you, and we have theories about laws. Laws happen in nature on our planet, when we're down here on planet Earth, these laws don't change. If you go out into space and when you learn higher level physics, you will know that some of these laws, for example, of gravity, don't hold true. But while we are on planet Earth, we have some laws of nature that are always true. They just happen. And then we come up with theories to learn about those laws, and we test our theories with a hypothesis. So make sure you can tell the difference between law and theory. You will for sure have a test question on that. All right. Tentativeness. This is your golden ticket for getting away with anything in science because we don't know. Everything that I'm teaching you, unless I tell you what a law of nature is, everything I'm teaching you is subject to change. Things that I learned in college are different than what I'm teaching you now because we have learned about them. I also want you to know what the fancy term empirical evidence means, which sounds really cool. It's basically just evidence gained through an experiment. It can be something you observe or something you actually measure. So it's basically a fancy word for all types of evidence. All right, here we go, scientific method. You need to know the steps of the method in order. Not a huge deal, but it's easy to remember that way. Everything that's in red is important, and you need to be able to not only know the steps, but know what they are and how to find them. So first you have to have a problem. There's no point in doing an experiment if you don't have a problem to solve. So in our class at the beginning here, I am giving you a problem of how do you make a radish plant grow taller. So that is your challenge. Good problems are usually why or how questions, all right? Then you form a hypothesis, which you know is an educated guess. So you're going to do some research on what helps a plant grow in general, and then you're going to make a hypothesis. A hypothesis is kind of like placing a bet. So I bet if I add vinegar to the water when I water my plant, then my plant will grow taller. So you're going to do some research, and maybe you decide that the chemical acetic acid helps a plant grow. I don't think it will. Maybe. I don't know. I wouldn't do it. Anyway, that's going to be my pretend educated guess. More on hypothesis. A good hypothesis is easiest to write as an if-then statement. So I would say, if I add vinegar water to a plant, then it will grow taller. My independent variable is what I'm questioning. And if you notice here on the screen, if I independent, I'll start with the letter I, and it comes right after there. This is what you are testing. It's what your education taught you or your research. How will you know if that independent variable is effective? You're going to get some data. So the dependent variable is what changes depending on what you're testing. So D for dependent, D for data, D depends on what happens here. Make sure you know the difference. So there's some more detail on how to remember that. I would write that red stuff down. Pause the screen. Okay, there are two types of data we can collect. Quantitative, think of numbers, or quantity. It is the best type of data because numbers don't lie. If your plant grows 5 inches, it grows 5 inches, although we're going to use centimeters metric. So if it grows 5 centimeters, then you can't deny what a scale says. That means it should be objective. It's based on an object, something that is unbiased. You could bias the growth of your plant. Maybe when you read the meter stick or ruler, you're going to kind of subconsciously round up. So there is bias in everything, and we'll talk about that more later. But quantitative data should be objective and unbiased. Qualitative data, think quality or opinions, and that is usually based on what you think. So maybe you think your plant looks really healthy. Maybe you think it's a nice bright green. Well, what does bright green mean to you versus what it means to me? What does healthy mean to you versus me? So these are more adjectives based on opinion, subject to change because of how we all think differently. So make sure you know the difference between the both. Make sure you know quantitative is better. Another look at objective versus subjective data. I do want you to know the difference between those words. Objects are a thing, subject is an idea. Okay, so maybe that helps. I could say that this is a rock. Yes, it is a rock. We could test it and prove it. Then I could say it's a cat-shaped rock. Well, that's my opinion that it looks like a cat. So be careful about giving subjective empirical evidence. You can read that if you need some more help on the difference between object versus subject. Make sure you know it. Okay, so now we have a problem, we have a hypothesis, we're going to run our experiment, and there are two important groups in an experiment. You have your experimental group, that's the one that actually has the independent variable added to it, and you have a control group, which is a norm. So in our plant example, if you think vinegar is going to help your plant grow taller, the plant that you add vinegar to becomes your experimental group. Well, how do you know the vinegar is going to work? You're going to have a control group. So your control group is the one that doesn't have vinegar. It's something that would be normal or a standard for comparison. So a good way to think of a control is this is what happens in nature. Vinegar doesn't rain down from the sky. I mean, yeah, it does. Acid rain a little bit, but that's not acetic acid. It's carbonic acid, in case you care. But normally a plant would grow with just plain old H2O, some dirt, and some sun. So your control group is normal, experimental group has your independent variable, and by having that control, it gives you more proof if your experimental group did anything. Maybe both of these plants grow the same height, so then you can infer that your experimental independent variable did nothing. Maybe your experimental group dies, so now you know that your acetic acid or vinegar killed the plant, or maybe it grows better. But if you don't have anything to compare it to, you can't really say that it grew taller or shorter or whatever. I'd rather talk about placebos in real life, so let's try to talk about that in class. And it's actually super cool, so help me remember. Constants in an experiment, what's kept the same? When you set up your plants, you want to make sure that the only difference between a control group and an experimental group is your independent variable. So the way you do that is you make sure everything else is the same. So when you plant these plants, they will be in the same size pot with the same amount of dirt. You plant the seeds to the same depth. You use the same type of seed. They get the same number of hours of daylight, the same amount of liquid. And in this case, one liquid would be water, one liquid would have vinegar in it or straight vinegar, but that would be the only difference. So then you can say for sure, if your plant grows taller, the only thing that would make it grow taller is the one thing you have different. So constants are super important in an experiment. Because these two words start with C, students often mess them up on a test. So control is your norm. Constants are things that don't change. So don't read things too quickly. Just be careful so you don't mess that up when I ask you about it. Okay, again, if you write a good hypothesis, you can figure out the independent variable, the dependent variable, and the problem. So, what are the effects of animal-based protein on blood glucose levels? So maybe your problem is, what affects blood glucose levels? Can't say that word. So then you would say, well, I think the amount of animal-based protein affects blood glucose levels. Blah. So your independent variable is how much animal-based protein you eat, and then your dependent variable is when you measure how much glucose is in your blood. If I drink a Monster, I will run faster. Independent variable is drinking that nasty stuff. Data is you're going to time a race. So you have to read between the lines a little bit and make some inferences so that you know what their data would be. Here is another hypothesis. It's not if then. How does the amount of nitrogen affect plant growth? What is my problem? How does a plant grow taller? What's my independent variable? If I add varying amounts of nitrogen, then the height of my plant or the growth of my plant will be affected. So you don't have to have an if then, but if thens are super easy to pull apart. If I independent variable, then data, track erase. All right, after we do the experiment, we analyze our data. Charts and graphs are super easy. If you have a good independent variable, those most of the time will be your x or your bottom axis. I remember that because a baby can lay on the ground independent of anybody's help. You don't need anything to stand on to lay on the ground. So your x-axis is most of the time your independent variable. Your data will go up and down the y-axis. So then you can see a rate of change as you change your independent variable. If time is a factor, usually days of the week or months of the year or hours or minutes go on the x-axis. And then if you're using time as part of your experiment, it's probably because you don't have lots of experimental groups. So I'll talk about that more in class, but being able to make a good graph is the best way to collect data. All right, at the very, very end, super important, you draw a conclusion, but real scientists communicate results. And this is where we write our lab report. So in order to truly do a scientific method, you are also writing a lab report, which, lucky you, is what we're going to do at the beginning of the year here. All right, in your conclusion, you need to say if your hypothesis was correct. If it was correct, you have not proved anything. You may have gotten lucky, so you need to test that over and over and over again to actually prove you're right. If your hypothesis is incorrect, then you rewrite it, and you are going to restart a new experiment. So when you communicate these results, you cannot say that you have created a new theory or proved your theory until you retest it. If you're wrong, you need to make a suggestion based on what you've learned from your lab of what a better idea would be. So we will do all that as we do this lab. All right, I'm going to ask you this on a test. What makes a scientific method valid? It is valid because it is repeatable. I can follow your procedure and redo your experiment. I can collect quantitative data so that I have unbiased empirical evidence to prove your hypothesis. That means that your data is objective and not subjective, even though we're going to include some opinions in there. And that means it's quantitative, and because of all that, it should be free of bias. We are all biased, whether you think you are or not. I'll talk about that in class, but you need to be careful. Okay, that is it for your first set of notes. See you in class.